Somewhere in the frozen soil of Siberia, in the sun-baked caves of Spain, scientists pulled DNA from bones that hadn't seen daylight in over 40,000 years. And buried inside that ancient code was something nobody expected: the fingerprint of a blood type so rare, so strange, that for decades no one could fully explain where it came from. Today, we finally can, and the real answer is far more fascinating than any myth built around it. Blood has always carried more meaning than biology alone. Long before anyone understood genetics, cultures across the world treated blood as something sacred, tied to lineage, destiny, even divinity. Royal families spoke of noble blood. Religious texts spoke of covenants sealed in blood. So when modern science finally identified distinct blood types in the early 20th century, and later discovered that some of those types behaved in unusually rare and medically significant ways, it's no surprise that old instincts kicked back in. People didn't just want to know the mechanism, they wanted to know the meaning. And that hunger for meaning is exactly what opened the door for so much speculation to rush in before the real answer was ready. Let's start with what most people think they know. O-negative blood is called the universal donor. In an emergency room, when there's no time to test a patient's type, doctors reach for O-negative because almost anyone can receive it safely. It's the blood used for trauma victims, for newborns in crisis, for soldiers on battlefields when there's no lab nearby. Fewer than 7 out of every 100 people on Earth carry it. In some countries, it's even rarer than that. That scarcity, combined with how quietly powerful it is in medicine, has made O-negative blood one of the most talked about and most misunderstood traits in the entire human genome. For decades, that mystery bred speculation, because O-negative doesn't behave like most other traits. It doesn't cluster the way you'd expect if it were just a random leftover from our shared ancestry. It shows up at unusually high rates in specific, isolated populations, most famously the Basque people of northern Spain and southern France, a group whose language and ancestry have puzzled linguists and geneticists for generations. Because of that pattern, alternative theories started to fill the gap left by hard science. Some say Rh-negative blood is proof of a hidden bloodline stretching back to biblical figures. Others claim it marks descendants of a lost, separate branch of humanity that never fully merged with the rest of us. And, in the more extreme corners of the internet, you'll find claims that O-negative carriers are the genetic remnant of ancient extraterrestrial visitors, engineered thousands of years ago and hidden in plain sight ever since. It's easy to see why these stories spread. Mystery invites explanation, and when science doesn't immediately offer a clean answer, people fill the space with something more dramatic. But here's the thing. Ancient DNA research, actual peer-reviewed genetic analysis pulled from real skeletal remains, has been quietly working on this question for years. And what it found doesn't need aliens, lost tribes, or secret bloodlines. It needs something far more grounded and, honestly, far more astonishing once you understand it. It needs deep time, migration, disease, and the sheer improbability of small mutations spreading through small groups of early humans wandering across a changing planet. To understand where O-negative blood actually comes from, you have to split it into 2 separate stories, because O-negative isn't one gene. It's 2 entirely different traits that happen to show up together. The first is the O blood type itself, part of what's called the ABO system. The second is the Rh factor, the negative or positive part, which is a completely separate genetic switch. Most people don't realize these are unrelated systems that just happen to combine in different people in different ways. Understanding each one separately is the key to understanding the whole mystery. Let's start with the O in O-negative. Blood type is determined by molecules that sit on the surface of your red blood cells, tiny markers your immune system uses to recognize what belongs in your body and what doesn't. If you have type A, your cells carry the A marker. Type B carries the B marker. Type AB carries both. And type O carries neither because of a very specific, very small genetic accident. Somewhere far back in our evolutionary history, a single letter of DNA was deleted from the gene responsible for building the A and B markers. That one small deletion breaks the instructions the gene relies on, so the marker never gets built at all. No A, no B, just a blank surface. That blank surface is what defines type O. Here's where it gets genuinely surprising. Genetic dating techniques suggest that this deletion, the mutation responsible for type O blood, isn't new at all. It's estimated to be roughly 1 million years old, far older than modern humans as a species. That means the mutation responsible for O blood type didn't originate with Homo sapiens. It was already circulating in the ancestral populations we descended from, and possibly in populations that existed before our species even split off. When scientists sequenced ancient DNA from Neanderthal, remains found in the Altai Mountains of Siberia, they discovered something remarkable, that Neanderthal individual also carried the O blood type. Our closest extinct relatives, a separate human lineage that diverged from ours hundreds of thousands of years ago, carried the exact same genetic quirk we still carry today. That single detail rewrites the entire story. It means type O blood isn't some late-stage evolutionary novelty unique to modern humans. It's ancient, inherited from a shared ancestor so far back in time that it predates the split between us and the Neanderthals. In evolutionary terms, A and B are actually the older, more conserved blood types, shared broadly with other primates like chimpanzees and gorillas. O is the newer variant, a disruption to that older system, and yet somehow it became one of the most common blood types on the planet. And in some populations, like indigenous peoples of the Americas, it became almost universal. Now let's turn to the second half of the mystery, the Rh factor, because this is where most of the strange folklore actually concentrates. The Rh system gets its name from rhesus monkeys, the species in which the antigen was first identified back in 1937. If your red blood cells carry this particular protein, you're Rh positive. If you don't, you're Rh negative. About 85% of people worldwide carry the positive version. The remaining 15% roughly do not, and that absence is what creates the second half of the O negative equation. It's worth remembering how recently we even understood any of this. For most of human history, blood transfusion was a dangerous gamble. Physicians experimented with transferring blood between people and sometimes between animals and people centuries before anyone understood why. Some transfusions worked and others caused patients to die suddenly and inexplicably. It wasn't until the early 1900s that the ABO blood groups were properly identified, explaining why mixing incompatible blood types triggered fatal immune reactions. The Rh factor came even later, identified in the 1930s, filling in another piece of a puzzle that had confused and endangered patients for generations. Once both systems were mapped, blood transfusion went from a life-threatening gamble to one of modern medicine's most routine, life-saving procedures. That history matters, because it shows how much modern confidence around blood type is actually quite young, barely a century old, compared to the million-year-old mutation sitting underneath it. Medically, this distinction matters enormously, especially during pregnancy. If a mother is Rh negative and her baby is Rh positive, inherited from the father, her immune system can potentially recognize the baby's blood cells as foreign material during birth or in later pregnancies. Left unmanaged, this immune response, called hemolytic disease, can seriously harm the developing baby. It's a real, well-understood, and now very manageable medical condition, thanks to treatments developed in the 20th century. But for a long time, before modern obstetrics fully explained this mechanism, this reproductive risk fueled speculation. If Rh negative blood behaves so differently, so incompatibly, some reasoned maybe it came from somewhere else entirely. Maybe it wasn't fully human. That gap in understanding is exactly where the myths took root. But ancient DNA tells a very different, very human story. Researchers studying genetic material from early modern humans and Neanderthals living across Europe and Asia, dating back between roughly 16,500 and 46,000 years ago, mapped out the blood types present in these ancient populations. This wasn't a small or narrow study. It looked at genetic variation tied to immune function and blood chemistry across multiple ancient sites, tracing how these traits shifted as populations moved, mixed, and adapted to new environments. What they found reshapes how we think about ancient migration itself. Think about the actual process behind this kind of research for a moment, because it's genuinely remarkable on its own. Ancient DNA doesn't come out of the ground clean and ready to read. It's fragmented, degraded by tens of thousands of years underground, contaminated by soil bacteria, and often present in vanishingly small quantities inside a single fragment of bone or tooth. Researchers extract what little genetic material survives, sequence it using techniques sensitive enough to piece together a readable genome from scraps, and then cross-reference specific genes, including the ones responsible for blood type, against known modern variants. It's a slow, painstaking process, and it's only become possible at this scale within the last couple of decades, as sequencing technology finally caught up with the questions scientists had been asking for generations. What makes the resulting data so valuable is that it isn't inferred indirectly from modern populations and then projected backward. It's a direct read of the genetic material actually present in people who lived and died tens of thousands of years ago. That distinction matters enormously. Instead of guessing what ancient blood type distribution might have looked like based on statistical models, researchers can now literally see it, sample by sample, site by site, confirming or overturning assumptions that had gone unchallenged for decades. As early humans moved out of Africa and into new environments across Europe and Asia, they encountered entirely new sets of pathogens, different climates, different diets, different exposure to disease. Blood type isn't just cosmetic. The proteins on red blood cell surfaces interact directly with the immune system, and different blood types offer different levels of resistance or vulnerability to specific diseases. Some blood types provide better resistance to certain bacterial infections. Others may offer protection against particular parasites or viruses common in specific regions. As small groups of humans spread into new territories, natural selection favored whichever blood type combination gave them a survival edge against the diseases they were newly exposed to. This is a critical point because it explains why blood type distribution isn't uniform across the globe. It's not random, it's the fossilized outcome of thousands of years of disease pressure, migration bottlenecks, and small founding populations settling into new regions. And when a small group of people move into an isolated area, whatever blood types happen to be present in that founding population become disproportionately represented in their descendants, especially if that population stays relatively isolated for a long stretch of time. This is called a founder effect, and it's one of the most well-documented phenomena in population genetics. This is exactly what likely happened with the Basque population. The Basque region has one of the highest concentrations of Rh-negative blood type in the world, alongside a language that doesn't fit neatly into the broader Indo-European family tree surrounding it. For a long time, that combination, unusual blood type frequency plus unusual linguistic isolation, fed theories about a separate older lineage of humanity surviving in a hidden pocket of Europe. But ancient DNA studies increasingly point to a more grounded explanation. The Basque region maintained a relatively isolated population for thousands of years, shaped by geography, mountains that limited large-scale migration, and mixing with surrounding groups. That isolation allowed certain genetic traits, including a higher frequency of Rh-negative blood, to become concentrated over many generations, the same way an isolated island population might develop unusually high rates of a particular trait simply through limited genetic mixing rather than external exotic origin. Similar patterns show up in other regions, historically associated with unusually high rates of Rh-negative blood, including certain Berber populations in North Africa and Bedouin communities in the Sinai Peninsula. In every case, when researchers dig into the actual population history, geographic isolation, small founding populations, and genetic drift explain the pattern far more convincingly than any story involving lost civilizations or non-human ancestry. It helps to picture how founder effects actually work in practice, because the math behind them is simple, even if the outcome feels dramatic. Imagine a small group of 50 people settling a remote valley cut off from surrounding populations by mountains or geography, staying largely isolated for dozens of generations. If, purely by chance, a handful of those founding 50 happen to carry the Rh-negative variant, that percentage doesn't stay static. It ripples forward through every subsequent generation, especially in a population that isn't regularly mixing with outsiders. Over centuries, a trait that started as a modest minority within the founding group can become dramatically overrepresented compared to the wider region around it, not because it was selected for, not because it conferred some hidden advantage, but simply because of how small, isolated populations propagate whatever genetic material they started with. This is the same basic principle geneticists use to explain unusually high rates of certain inherited conditions in other isolated populations around the world, from island communities to religious groups that historically married within their own community. There's nothing mystical about it. It's just genetics playing out exactly as you'd expect, given enough time and enough isolation. Now, let's directly address the extraterrestrial theory, because it deserves a clear, honest answer rather than dismissal. The core claim usually goes something like this. O-negative blood is described as lacking a specific protein marker that positive blood types carry, and some theories suggest this absence indicates non-human genetic engineering, ancient intervention, or hybrid ancestry linked to visitors from beyond Earth. It's a compelling narrative, cinematic even. But it runs into a wall the moment you apply basic evolutionary biology. If life exists elsewhere in the universe, and it very well might, there's no reason to assume it would share our genetic architecture at all. DNA itself is one particular molecular solution to storing biological information shaped by billions of years of chemistry unique to Earth's early environment. An organism that evolved on a different planet, under entirely different chemical and environmental conditions, would likely use a completely different information storage system, if it uses anything resembling genetic code at all. The idea of a viable biological hybrid between humans and a truly extraterrestrial organism runs into the same problem famously described by astronomer Carl Sagan, who noted that creating a viable hybrid between two organisms requires a shared underlying biology, and crossing something truly alien with a human would be less like breeding two related species and more like trying to cross a human with a tulip. The biological machinery simply wouldn't be compatible. So when you strip away the dramatic framing, what you're left with is this. Every trait involved in O-negative blood, the ABO deletion and the Rh-negative variant, has a clear, traceable genetic mechanism. Both mutations exist within known human and pre-human genetic history. Both can be dated using standard techniques used across the field of population genetics. Both show up in ancient remains that predate any supposed period of ancient extraterrestrial contact. Remains tens of thousands of years old in some cases, remains that predate our species entirely. Occam's razor, the principle that the simplest explanation consistent with the evidence is usually the correct one, points overwhelmingly toward ordinary evolutionary processes, not intervention from beyond our planet. That doesn't make the real story boring. If anything, it makes it more remarkable because what ancient DNA actually reveals is a genuinely epic story about human movement and survival. Picture small bands of early humans, no more than a few dozen individuals at a time, pushing north out of Africa tens of thousands of years ago. They're moving into unfamiliar terrain, encountering colder climates, new food sources, and pathogens their immune systems had never faced before. Some of those small groups happen to carry, by pure chance, certain blood type variants. Some of those variants happen to offer a slight survival advantage against local diseases. Over dozens, then hundreds, then thousands of generations. Those slight advantages compound, spreading through descendant populations, while other blood type variants become rarer or disappear from certain regional groups entirely. At the same time, some of these early human groups encountered and interbred with Neanderthals, another branch of humanity that had already been adapting to European and Asian environments for hundreds of thousands of years. That interbreeding wasn't a footnote. It left a measurable genetic legacy still present in people today, including immune system variants and blood-related genes inherited directly from Neanderthal ancestors. The ancient DNA studies that mapped blood types across ancient Eurasia found evidence of this exact kind of genetic exchange, blood group variants moving between modern human and Neanderthal populations as they crossed paths, mixed, and eventually as one lineage absorbed the other, blended into the ancestry of nearly everyone alive today with roots outside Africa. It's worth pausing on just how strange and wonderful that Neanderthal connection really is. For most of the 20th century, Neanderthals were imagined as a dead end, a separate species that competed with modern humans and eventually lost, vanishing without leaving much behind. Ancient DNA completely overturned that picture. We now know that when modern humans expanded out of Africa and encountered Neanderthal populations already established across Europe and parts of Asia, the two groups didn't just compete, they interbred repeatedly in multiple locations over a long stretch of time. Most people with ancestry outside Africa today carry measurable percentages of Neanderthal DNA scattered throughout their genome, remnants of those ancient encounters. Some of that inherited genetic material affects immune function. Some of it affects skin and hair traits. And some of it, as ancient blood type mapping has shown, is directly tied to the blood group variants still circulating in human populations today. Neanderthals weren't a separate failed branch of humanity that vanished into irrelevance. In a very real genetic sense, they never fully disappeared. They were absorbed, and pieces of them are still quietly present in blood type distributions around the world. This is the real secret ancient DNA revealed, not a hidden alien bloodline, but a record of an extraordinarily complex, extraordinarily human journey. Every time a small group of early humans pushed into new territory, faced a new disease, survived a harsh winter, or crossed paths with a different human lineage entirely, it left a mark, encoded permanently in the blood type frequencies we still carry today. O-negative blood isn't evidence of something added to humanity from outside. It's evidence of just how much pressure, movement, and adaptation shaped humanity from within over an almost unimaginable span of time. There's another layer to this story worth understanding, because it explains why O-negative specifically, rather than any other blood type, ended up carrying so much mythology. It comes down to a strange overlap of two separate systems, both of which happen to become rarer in certain regions for entirely unrelated reasons. The O blood type became dominant in certain founder populations, partly through genetic drift and partly through selective pressure from disease exposure. Separately, and for entirely different reasons tied to different genes on a different chromosome, the Rh-negative variant became concentrated in certain isolated groups through geographic isolation and small population size. When both of these independent, unrelated genetic patterns happened to overlap heavily in a few specific human populations, like the Basque people, it created the appearance of something unified and mysterious, a single unusual trait, when in reality, it's the coincidental overlap of two separate ancient stories. This is actually a really important lesson in how scientific mysteries get built. Humans are pattern-seeking by nature. When we see two rare things showing up together repeatedly, in this case, rare language isolation and rare blood type frequency, our instinct is to look for a single explanation that ties them together. Sometimes that instinct leads to real discoveries, but sometimes it leads us towards stories that feel satisfying without actually being supported by the underlying mechanism. Ancient DNA research is powerful precisely because it lets scientists move past assumption and pattern matching and instead directly test what genetic material from real ancient individuals actually shows. And what it shows, again and again across multiple studies, multiple ancient sites, and multiple independent research teams, is a consistent, traceable evolutionary history. The O mutation, dated to roughly 1 million years ago, present in Neanderthals as well as modern humans. The Rh negative variant, tracked through populations across Europe, Asia, and Africa, showing clear signs of founder effects and regional isolation rather than any external origin. Blood type frequencies shifting measurably as ancient populations migrated into new environments and encountered new diseases, exactly as you'd expect from ordinary evolutionary adaptation, not from any engineered or non-human intervention. So what does this mean for you if you happen to carry O-negative blood? Or if you're just fascinated by where it comes from? It means you're carrying a genetic signature that stretches back further than most people realize, a mutation older than our species, inherited from an ancestral lineage we shared with Neanderthals, combined with a second, genetic trait shaped by thousands of years of small, isolated human communities, adapting to their specific corner of the world. It's not evidence of something added to humanity from outside. It's a living record of how humanity spread, adapted, and survived, written directly into your blood. It also explains why O-negative blood remains medically valuable today, in an almost ironic twist of evolutionary history. Because the O blood type lacks the A and B surface markers, and because Rh-negative blood lacks the Rh protein marker, O-negative blood cells are far less likely to trigger a dangerous immune reaction when transfused into someone with a different blood type. The very same genetic absence that fueled decades of speculation and mythology is the exact same absence that makes this blood type so uniquely useful in emergency medicine today. In a strange sense, the thing that made O-negative blood seem mysterious for so long is the same thing that makes it medically remarkable. Its blank genetic canvas, inherited from deep in our evolutionary past, happens to make it compatible with almost everyone alive today. It's also worth remembering how much this changes the way we should think about mystery itself. Not every unexplained pattern needs an exotic explanation. Sometimes the explanation is simply that we hadn't yet built the tools capable of reading the answer directly. For most of the last century, questions about the deep origin of blood type variation had to rely on comparing mother and populations to each other and working backward through statistical inference, an approach that leaves plenty of room for gaps and plenty of room for speculation to rush into those gaps. Ancient DNA closed much of that gap by allowing scientists to read the genetic record directly from people who actually lived through the events in question, rather than reconstructing it secondhand from their descendants thousands of generations later. That shift from inference to direct observation is one of the quiet revolutions in modern science, and blood type history is just one of many stories it has rewritten. There's something worth sitting with here. For decades, uncertainty around O-negative blood got filled with increasingly elaborate stories, hidden bloodlines, ancient alien intervention, secret tribes untouched by ordinary human history. But the actual explanation, pieced together patiently, through ancient DNA analysis, population genetics, and careful comparison across tens of thousands of years of human and Neanderthal remains, turns out to be more compelling than any of it. Not because it's stranger than fiction, but because it's real, verifiable, and traceable directly through the genetic code preserved in bones that are tens of thousands of years old. What ancient DNA gave us wasn't a secret meant to stay hidden. It was patience finally paying off, decades of careful sequencing, comparison, and analysis, slowly filling in a gap that speculation had rushed to fill with something more dramatic. And in doing so, it revealed something genuinely worth marveling at, that a tiny genetic deletion, older than our own species, carried by Neanderthals in Siberian caves and by early humans crossing into unfamiliar continents, still lives on today in the veins of millions of people, quietly shaping who can safely receive blood in an emergency room, and quietly connecting every person who carries it to an unbroken chain of ancestry, stretching back nearly 1 million years. There's a certain irony worth appreciating too. The very features that once made O-negative blood feel foreign, feel almost too rare and too particular to be ordinary, are the same features that make it so deeply, unmistakably tied to the ordinary mechanics of evolution. A deleted letter of genetic code, a missing surface protein, small isolated populations, passing traits forward through generation. None of it is exotic in the way the myths imagined, and yet all of it, taken together, tells a story just as sweeping, just as improbable, and just as worth telling around a fire at night. The difference is that this version happens to be true, and every claim in it can be checked against real, physical evidence pulled directly from the ground. That's the real story hidden inside O-negative blood. Not aliens, not lost civilizations, not hidden bloodlines whispered about in forgotten texts. Something far older, far stranger in its own right, and entirely, unmistakably human. A mutation that outlived an entire other species of humans. a trait shaped by disease, migration, isolation, and chance, repeated across tens of thousands of generations, and a blood type that, purely by evolutionary accident, ended up becoming one of the most valuable substances in modern medicine. The next time someone tells you O-negative blood hides a secret extraterrestrial origin, you'll know the real answer. One written not in speculation, myth, or guesswork, but in the actual genetic code preserved patiently inside bones that have waited tens of thousands of years to finally be read, tested, and understood.